Hi and welcome everyone. FluGuide recently released a strategic update regarding the clinical development of lead candidate FG001, and with us today we have the company's CEO Morten Albrechtsen and the company's CSO and CMO Andreas Kjær to discuss the strategic update. Gentlemen, welcome and please go ahead with your presentation.
Thank you, Christian. Thank you for the introduction. Welcome everyone. So we are pleased to present an update we have done in some of the priorities, and we'll go through some of the rationales. We start out with some of the background that will lead up to explain why we do make the choices we do. FluGuide, as you know, is working on precision surgery for cancer patients, and please make your own assessment. You know, it's unfortunately 80 million patients every year have cancer. 80% of them undergo surgery, and half of them will not have a sufficient surgery in the sense that it could recur after surgery. This is because the surgeon is not yet able to see all the cancer. They can only feel it, and they see it with their eyes. We help them to see more cancer and do more precise surgery. So the way it works, we have this image agent, FG001, that is reconstituted, it's injected into the patient prior to surgery. It will circulate, bind to cancer, and then it's ready during surgery for the surgeon to actually see it. There's also features that we'll come into with FG001 that is quite interesting and make some of the reason for why we have made the priority changes we have done. So FG001 is a product that consists of a peptide that bound to UPAR, and they have the UPAR specific characteristics. Andreas will tell more about that. But it's also the coloring molecule, the ICT, is very important. It's known to be extremely well tolerated. It had the same spectral specifications as ICT, meaning that it works on the same equipment. So if an equipment can see ICT, it can also see FG001. And then there's a second generation product that you can see deeper into the tissue. You see the two images. Andreas will also actually give more data on that. But on the right side, you see the cancer with the near-infrared light. You don't see it with the visible light, and that's a key benefit of near-infrared light. So the summary of what we do now on the refinement of the strategy is really that we was blessed with last year to have 3 phase 2 results that was all extremely good. We have it in brain, aggressive brain cancer, lung cancer, and head and neck. And well, we wanted to prioritize. We cannot move ahead 3 programs in parallel. And what you will see is actually each indication consists of a couple of applications. So just one indication, not just one product. Actually, there's multiple products underneath and multiple opportunities commercially for helping the patients. So actually what we do, we focus in on 2 of them. Is the brain and head and neck for now. I'll explain why later. We look to the full thermotherapy, which is an treatment. It's not only guiding the surgeons during surgery, but actually is also helping the surgeon to treat the patients. And then last but not least, we have the head and neck that consists of multiple different applications that can help patients depending on where the head and neck cancer is located, what problem the surgeon has, and that gives us opportunity both for having multiple products that help head and neck cancer patients, but also to do multiple partners that we can carve out the different aspect, and we can then sell, so to speak, it multiple times. Andreas, it's yours.
Yeah. So I'll take over, and just for some it might be a repetition, but just remind you about the beauty of the UPAR targeting technology and why we believe it's very strong. It's the target, which is the handle we bind to UPAR, is expressed on the receptor surface of cancer cells. And it's actually a marker of the metastatic process. It's involved in breaking down surrounding tissue. So for that reason, it's the more positive it is, the more UPAR there is, the more aggressive the cancer is, which makes it really good because the more it's needed, the more our technology will work. It's expressed at the invasive front of the tumor. What does this mean? It's actually highly expressed at the surface where we want to delineate the tumor. So this makes it a very good marker for the surgeon to know what to remove and not to remove. And more than 200 publications, not with FG001, but with UPAR and cancer have been published. So it's on a very solid ground we are building our technology. The other very good thing about the technology is that it's expressed in more than 80%, in many cases more than 90% of solid cancers. What does this mean? It means that once we have developed the product, as we have, FG001, we can use this in a variety of cancers, basically anticipated in most solid cancers. We've now tested it in 3 different cancer types and are pursuing in particular 2 of these, but the field is open. It only depends on the bandwidth, how much we can manage and where we believe the unmet need is the largest. And if you take the next one. So how does it look in real life? This is how it looks. If you look to the left, this is tissue removed from a tongue cancer, so a cancer sitting in the tongue. And to the very left, you see how this is taken out of the patient, and then you can see the orange part, orange-blue part, is where the tumor sits, but the rim outside, where it's not marked, is normal tissue. So this demonstrates that the surgeon got all the tumor removed. If there had been edges where the signal would have been present, then the surgeon would have to go back and see, well, here it seems that I didn't get negative or normal tissue around the tumor, and there might have been left some cancer in the patient. And that translates to a poor prognosis. If we look at it more microscopically, you can see a slice, a thin slice of a tumor. And to the left, you see how the tumor lights up in green here. That's FG001. You can see in the middle, the yellow line is the pathologist drawing where the tumor is. And on the right, you see the brownish color, which is where UPAR is. So this is an exact fingerprint of where the tumor is. We have the UPAR expression and we have the light signal. So if you remove guided by the light signal of FG001, you are actually delineating the tumor correctly and making sure that we remove all of the tumor. The next slide. But another layer, and that's something we are going to talk about today, and I'm sure also discuss afterwards, is that not only can we guide the surgeon by FG001 because ICG lights up, so that's the images you just saw, but actually the compound has other properties. And one of these properties is that if you light on it with a laser with relatively high energy, that energy is absorbed and converted into heat, and heat kills cells. So we can apply this for what is called photothermal therapy by lightening on it and demonstrating that we kill cells. And we have done this in animal models of human cancer and demonstrated that not only do we kill cancer cells and not normal tissue, but we also increase the survival of these animals. So this is extremely promising. We are very excited, and the potential is that we can actually do a 2-step procedure with our compound, first using it for removing the cancer, but afterwards removing or irradiating with heat, removing those few cancer cells that might have been left behind. You could say we clean further up. And this is extremely exciting. And this is because we have selected the fluorophore in a wise manner that we can do this. Many fluorescent compounds that can be used maybe for guiding surgery do not have this capability. So this is why we want to go deeper into whether this can be an added benefit of our technology. If you take the next. So as I mentioned, we are in 3 indications: brain, head and neck, and lung. And the top 3 green bars are all on the fluorescent guided surgery. So FGS, the lightening up and guiding the surgeon. And the Second from the bottom, brain cancer PTT is the photothermal therapy. So the heating up once the compound is in there. And of course, maybe if I didn't explain that very clearly, the idea is, of course, that because FD001 sits on cancer cells, but not on normal cells, you are only heating up the cancer cells. So it's a gentle way to remove cancer. Then we are pursuing mainly brain and head and neck right now, but we also had positive data in lung cancer. But we dig more into the data we have in the two first indications. And then finally, we have also another compound that we will pursue in due time, but this is not really where the focus is right now. If you take the next.
Yes, so just highlighting the need in the market, more details, and also the market and the numbers. So in brain cancer, we're talking very much on aggressive brain cancer. Glioblastoma or high-grade glioma has different names. And if we look at it, the problem first. So the problem for the surgeon is always, do I remove more tissue from the cavity, or do I leave tissue behind? If they remove more, the risk to disable the patient, and if they remove too little, the risk that the cancer will regrow. So this is a situation, all types of cancers that will ultimately kill the patient, and cancer that is non-malignant, and that the patient will live with their entire life. So the two types of cancer, the malignant one and non-malignant one. The malignant one, the key problem is that in a year, approximately half of the patient will die, unfortunately. So the key focus on the malignant cancer is to remove all cancer and get the patient to survive. For the non-malignant cancer, actually only 10% of the patient in 10 years will have a recur of cancer, but 20% of them will have side effect from the treatment. And there the problem is much more to avoid the side effect and be able to do the surgery as precise as possible. So the solution, regardless if it's malignant or non-malignant cancer, is for all of them, and that is that the surgery has to be done precisely. And if you look at the numbers, I mean, this is just to give you an idea of where we are. I mean, what we look into with high-grade glioma is in fact only the malignant cancer that come primarily from the brain. And of those, as you can see in the figure, is a dark green, small 15% of them is only what we're looking at. But there's double as many primary, cancer coming from the brain, malignant cancer in the brain. And then there is 2 times as much non-malignant, where mini-glioma is doing half of them. So there's unfortunately a lot of different kind of brain cancers, but we just focus in on high-grade glioma as the first one. If you then look at metastases that come from other cancers to the brain, the number is actually quite high as well, unfortunately, and those also undergo surgery. So I think it's just an important point that fluorescence-guided surgery of brain cancer is much more than high-grade glioma. We, of course, have the focus on high-grade glioma, but it's an interesting aspect that they are all treated by the neurosurgeon. So they mean they all have the same equipment, they're all the same surgeons, it's the same customer group, and they're quite small in numbers, but they are huge in impact. So I just want to tell you this. I mean, there have been some questions about what is the size of the market, what's the size of the segments. But in the brain cancer, we go for the aggressive brain cancer first, and then later on we will branch into other cancer potentially. But it's unfortunately a high number of patients have these cancers. And then I'll hand over to you, Andreas.
Yeah, and I'll go a little bit into the data. And I think just Morten, to follow up on the last thing you said, I mean, ideally we could see a scenario, but it remains to be proven, where the neurosurgeons could basically use FD001 for any brain tumor surgery. So, but of course that needs to be shown in individual studies in the different indications. But high-grade glioma is the starting point. And that's what I'm going to talk about, where we have completed 2 studies, a phase 1/2 study and a phase 2 study. And we have reported the top-line results previously of these, but now we'll just give you an overview of how strong these data are and why we're very excited about it moving forward, and hopefully being able to help a lot of patients with this very aggressive cancer disease. So the patients in both studies were patients suspected with high-grade glioma undergoing surgery. And the phase 1/2 study included 40 patients. And as always, in an initial study, the primary endpoint was safety and tolerability of FG001 and finding the right dose. The right dose means the dose that gives the best signal without having if once we are not gaining more by increasing the dose, of course, we are not going higher in dose. The drug that was given here, we compared with 5-ALA because that is a compound that is used currently by some surgeons to guide this brain cancer surgery. And what we did was that we co-administered it to all patients. So all patients had both compounds. Does this make sense? Yes, because you use different wavelengths, so you do not see the two signals at the same time. So you can look at it with FG001, and then you can switch, and then you can look at how it looks at 5-ALA. The strength of this design is that the variation between patients is removed. It's exactly the same patient you see. And as you see, for instance, the images to the right, I come back to those, it's not comparable patients, it's the very same patient. So you can one-to-one, head-to-head, compare how it works. We did dose escalation from 1 to 48 milligrams, and from 36 to 48 milligrams, the two highest doses, we didn't gain more. So then we stepped back to 36 milligrams. That was then the dose that was used in the phase 2 study. And the phase 2 study was made in a little bit different way. Now it was 24 patients, but now we randomized patients. So now the patient did not get both compounds, but they were randomized into either getting FG001 or 5-ALA. 12 patients each. And primary endpoint now was at least one indeterminate tissue or unexpected fluorescent tissue at the end of surgery. This is very complicated to understand, so I'll try to explain it a little bit more simple. Basically, the readout was the surgeon did the surgery in white light, the traditional way to do it, and said, I'm done. Now I moved all of the tumor. Then the light was turned on, so one could either see FG001 or 5-ALA, and then the surgeon, actually I can already tell a little bit from the next slide. In all cases then the surgeon saw extra tissue. That tissue was removed and tested whether it was actually cancer. So how large a percentage of the patient had more cancer removed due to using the optical guidance. So the results were that all patients receiving FG001 had additional cancer detected. So we did something good for the patients, at least on removal of cancer. More cancer was removed, or you could say cancer was overlooked when using white light. And that was in 100% of the cases. This was the same for 5-ALA. And there was no statistical difference between the two in accuracy of gross total resection. And FG001 also lit up in all patients. And this is when we, not only in the phase 2 study, but also in the phase 1/2 study, 48 of the 64 patients lit up. Why is this not all of them? It's because we started out with 1 milligram, very low dose, and then we increased the dose. But from 8 milligrams and upwards, they consistently lighted up. So very robust. And also a large margin from the 36 milligrams we use. So if one patient is larger, so the dose in theory is a little bit lower per kilogram, we still have a lot of margin down to only having dosed 8 milligrams. That would still have been sufficient to see the tumor. It was safe and well tolerated. with only 2 related, very minor side effects. And in comparison, there were actually 10 related adverse events in the 5-ALA group. So more adverse events in that group. And then, as Morten briefly mentioned in the beginning, our technology can see 1 to 2 centimeters into the tissue, whereas 5-ALA can only see 1 to 2 millimeters into the tissue. And that is why the 2 images of the same tumor, it's the surgeon that had opened and gone through the dura and is now on the surface of the brain. And on the left, the surgeon can already see in depth where the tumor lies, whereas with 5-ALA, the surgeon cannot see that. That is only once the surgeon starts to do the dissection. And why is this valuable? It's valuable because for the planning and where the surgeon starts the surgery, it's good to be guided beforehand. So it's a clear advantage of the technology. Yeah. So again, phase 1-2 key results, outstanding safety profile, optimal dose found with 36 milligrams, where there's a high contrast, and we went all the way from 1 milligram up to 48, so we know very precisely where the correct dosing is, and it has a broad window. And then we demonstrated by histopathology that that was tissue samples that light up contain cancer. We didn't want areas to light up that were not cancer, because then, as Morten mentioned previously, one thing is removing all the cancer, but the other part is only to remove what is cancer, because there are a lot of important functions in the brain you don't want to remove if it's not cancer. And this is again the primary endpoint in the phase 2 study, so the one where we randomized to the 2 groups, where the endpoint was, did we see more cancer that was removed only by using the optical guidance? And yes, we did this in 100% of the cases, so as good as we could have hoped for. And this is just a video demonstrating that when we are looking at biopsies, we took 31 biopsies in this study, so more biopsies from each patient. How could we compare whether it lighted up and whether it contained cancer? And that was by white light you see on the left and the optical signal you see on the right. So you can see areas where it's pointing at where it's lightening up, and adjacent to that areas where it's not lightening up. So you take biopsies from areas that are positive on FD001 and areas that are negative, and then you compare the histopathology, and that is whether the pathologists say, yes, this is cancer tissue, or no, this is normal brain tissue, or not cancer. Yeah. So the deep visualization, again, this is just a close-up of it. You can see this patient lighted up. You could see where the cancer is, not with 5-ALA. And if we take this across all the cancers, not surprisingly, none were visualized by 5-ALA. Remember, it's 1 to 2 millimeters depth. And we saw it in 4 out of 12. So in 1/3 of the patient, we saw it. You might ask, why are we not seeing it in all patients? That is because some of the tumors are not located adjacent to the surface. So even the 1-2 centimeters we can look into it will not visualize it, because it might lie in the very middle of the brain, maybe 5 centimeters from the surface. So we didn't expect to see 100%. But if it lights up, it definitely guides the surgeon. That's the feedback we get from the neurosurgeons. And also the details on the related adverse events, very few and only mild, what is called grade 1, and substantially more with 5-ALA. In 5-ALA, it's mainly nausea and vomiting that is a known side effect. And the accuracy, so this is the samples where we took 31 biopsies. You saw the video, and then it was matched to whether it was cancer or not. And again, positive predictive value, which means if it lights up, it is cancer. And you see all the very high numbers of sensitivity and specificity. Statistically, we are a little bit better if you look on the numbers, but because it's very few patients, statistically you cannot say or claim that this is better. But the raw numbers are very encouraging here, and very high specificity and positive predictive value, which is what we want. And then sometimes surprises happen during surgery, and this is actually a patient that turned out in the study to have a meningioma, which is another type of brain cancer, and this patient actually lighted up, and it was even at it was not 36 milligrams because it was in the dose escalation phase. So this patient got 8 milligrams, and as you can see within the yellow outline that has been put on where is the cancer in the upper right corner, you can see how it looks with FG001. So very encouraging. So we have preliminary data that we can actually see meningioma also. And if you think back to Morten's pie chart, how many cancers in the brain there are, this adds already another indication potentially, and more patients than just a patient with high-grade glioma. And by the way, meningioma is a specter, so they can be relatively benign, but the aggressive ones are very aggressive. So there's a big need also here to be able to remove the whole tumor and nothing but the tumor, basically. Yeah. So one of the advantages and why we're also pursuing high-grade glioma, apart from the very poor prognosis and the high unmet need and the real need to develop something that can help these patients getting a better prognosis and a better quality of life following surgery, is also that microscopes are already used routinely for these surgeries. And they come with optical capabilities, so they can actually see these fluorophores. And as Morten mentioned, ICG is a standard fluorophore. So we developed FG001 to make sure it was immediately compatible with the existing equipment. And Zeiss, Leica, and Olympus are some of the players in this field. So it's a very easy way into the operating theater and the surgeon's mind, because they already use this kind of equipment. On the photothermal therapy, where we apply also laser, but not just to get it to light up, but a little bit higher energy to convert it to heat, there are also several partnerships, potential partnerships, and several equipment and approved equipment for using this laser in surgery. So a lot of partnering opportunities. Yes. So we also had feedback from FDA based on the studies we did now. And why are you interacting with the FDA? That's in order not to get surprises later on, and to be guided on how to make your pivotal study, your phase 3 studies once you are there. And the feedback was extremely positive. What we were told was that safety needs a database of approximately 300 patients. That didn't come as a surprise. This is pretty standard, but we can pool this across the different trials we do. So already we have some of the patients in this, and the different trials will add up to this number. Then we got a feedback that completeness of resection seems an acceptable endpoint. So basically you look at, did you leave anything behind? Did you get everything out? And do you, in a more complete way, do you do your surgery more complete when you're guided by the optical signal. And of course, this is very likely, or maybe you could say almost a no-brainer, in the way that, yes, we have proven we see more cancer tissue by turning on the fluorescent signal, and that is then removed. So this will also supposedly lead to that the resection is more complete. And then they also suggested a follow-up of 6 to 12 months, which is a positive, because in theory this allows us to detect any benefit in the outcome, but it's probably not a prerequisite for an approval. If you can show you are more complete in the resection, that is probably the key readout. But the patient would be followed for 6 to 12 months. And remember, these patients, unfortunately, only have around 12 to 15 month median survival following the surgery. So it seems a short follow-up, but it's a long follow-up relative to the severeness of the disease. Yeah. So the plans based on the data we have and the FDA feedback is relatively clear to us. We want to initiate a phase 2 trial. That is another phase 2, assessing the potential of photothermal therapy in aggressive brain cancer, because we would see it as a missed opportunity not to include that already now, because the potential is so big that instead of doing it sequentially, first look at optical guidance and then look at photothermal therapy would almost be unethical if we turn out to show that PTT actually works in these patients and we already dosed the compounds. We think it's almost an obligation already now to think how we could implement this into a study. And then, of course, expanding support for reimbursement. If photothermal therapy works, that, of course, is a different game on reimbursement. And then expanding the usability. Part of this is we have a compound that can be flexibly dosed. So we already tested it the same morning, the day before, evening or morning on the day before. And it seems on the data we have that it's quite flexible when you can dose it. This is logistically an advantage, but it's also, for instance, if a patient is postponed from morning to afternoon surgery, it's not a problem with our compound, where the window for 5-ALA is much more narrow. So yes, so evaluate inclusion of photothermal therapy. Of course, obtain further FDA feedback on our intended design of a phase 3 clinical endpoint, because we want to test that endpoint already now, and then expand the documentation of safety and efficacy, and that is kind of automatically coming in every time we are doing further studies.
Yes, that was the brain review of the data and strategy. Then in head and neck, on the market and the way it works, actually head and neck cancer, they have cancer from a variety of different anatomical locations. Those that are on the upper part of the head and neck, I mean in the mouth and the tongue, they're most typical one, and they're the one that is easiest to get to. You can get to them with the open field cameras, and they are visible for the surgeon. The key problem from them is to check how much actually you take out. I mean, you don't want to take too much out, more than you need. And if you take out too little, then you leave something behind. So what they do, they check the surrounding of the cancers to see if they have get everything out. Unfortunately, today, the golden standard is the histology that is answer coming 14 days after surgery. They have some freeze section that can become earlier, but they're not very reliable. So they rely on this 14-day feedback. And at that time, the patient has been, I mean, the surgical wound have been closed and the patient has been dismissed from the hospital. And the only thing you can do thereafter is either to do a resurgery or you can give them chemoradiotherapy. So helping the surgeons to have an on the fly, so to speak, to have an answer in the surgical room that they are too close, that they can move more, would be a major benefit for those patients. So the patient then that have the cancer deeper into the head and neck, I mean, into the pharynx and oral pharynx, you cannot reach them really as easily, and you need some new tools to get into them. That could be a scope, that can be a robotic equipment, but you need something to get down to the cancers. Today, those patients will often just have radiochemotherapy. You not do the surgery because you cannot do it. So what we can do there, we can enable new equipment actually to be used in this indication, enable the patient to have another treatment modality added to the treatment, I mean surgery, and we can help the surgeon actually to play a higher role in this deeper located cancer. So that is difficult to reach kind of cancers. And what we need for all of them is that we need to have more precise surgery, and we need to be enabling to them to be closer market that they can do safely. So in terms of the market, the number of head and neck cancer, unfortunately, also is very high, and that's I mean, divided between the different kinds of the locations of them. I mean, we've been asked many times for the numbers. I mean, here it's the numbers, and all those cancer locations and say use situation that the surgeon have, a problem the surgeon have, we can help in different ways. And actually that means that we can promote equipment for different use. We can promote equipment that check the cavity in vivo. I mean, when they do the surgery, we can enable equipment used when they have done the surgery and check the surgery is done completed. And we can also enable new equipment to come into play in the deeper located pharynx. And that way, we'd actually do multiple partnership potentially. And we have multiple, let's say, products that we could document. And this is interesting at the head and neck contain almost all for what kind of applications that is important for a type of cancer. And using head and neck as a model, we actually cover some of the problems that will occur in almost any other cancers. So this is really a model cancer for us for all other cancers with equipment and also in terms of surgical techniques.
Yeah. So now to the data on head and neck cancer, where we performed a phase 2 study. The top-line results have also been communicated at high level previously, and the inclusion, the patients here were with oral or oropharyngeal squamous cell carcinoma scheduled for surgery. So pointing back to what Morten said, this is a subpart of head and neck cancer that are easily accessible, some of them easier oral at least, because we wanted to start out with something where we could look at the light signal. One of the interesting things here is actually if it's a tongue cancer, it's sitting there in the mouth, so you can actually have a look at how it lights up over time without actually starting to do the surgery yet. So we got a lot of information also for when is the optimal time window, how sensitive is it, and so on. But that was not the major endpoint. That was we included 16 patients, and it was the sensitivity. So could we see the cancers? Were the cancers lightening up? So as a proof of concept. And the drug was the same as in brain cancer, and we dosed it in 4, 16, and 36 milligrams per patient. And you could ask, why did we not just stick with the 36 milligrams? You can discriminate between brain tumors because there is what is called a blood-brain barrier, and then peripheral tumors like head and neck and most other cancers. So you couldn't be absolutely sure that you needed exactly the same dose, and that's why we went back to also look at different dosing. So we show relevant contrast in all patients, so regardless of the dosing. So it all worked fine, and it was safe and well tolerated in all patients. And then how it works, if you look at the image below, you cannot see anything. This is white light. And this was what the surgeon is seeing when checking for local metastasis, typically lymph nodes. So it may have spread outside the tongue or where the tumor is sitting. And then normally it's by palpation, so by using the fingers to feel where could there be something that is hard or enlarged. But once the surgeon turned on the near-infrared light, so FG001 showed up where it was located, and on the next slide, Morten, then this happened. So suddenly the surgeon could see, wow, there's sitting a metastasis right in the middle. You can see gripping around it, and this is a thing you cannot see. And this will lead to metastases being found that were not detected otherwise, because you could imagine that palpation with your fingers is not a safe procedure. You have to check everything through, and you cannot be absolutely sure it is a metastasis or not. But when it starts lighting up, you're really guided, and then you can go directly to that area. So extremely exciting and extremely valuable, because if you leave behind metastasis, the prognosis is very poor. Yeah, if you go to the next. So there's actually a range of potential uses of this, and this is again and again we are amazed about the luxury of having to select how to position the product. It's not like it's only working in one way, and we are now digging deeper into this. On the right, we have basically shown the 3 different scenarios where you can use it. One way to use it is a little bit in line with what is done with aggressive brain cancer, namely to check whether there's still cancer left behind. So when you can guide your surgery following the optical signal, but then you can look in the cavity, is there still sitting an optical signal? Then you clearly have left cancer behind. So this is one way to do it. Then there's when you take out the specimen in the middle bar, and as I explained in the beginning, then you have a specimen and you would like the rim to be negative for signal, because only then you are sure that you have obtained what is called negative margins, that you are not so close that you had left something behind. And in head and neck cancer, it has actually been proven that not only do you want to be clear of cancer, but you must also add some millimeters of what looks like healthy tissue, but have some hidden cancer cells in it to be sure that the patient is not relapsing, getting a regrowth of the tumor. And then finally, as shown on the video on the last slide, detection of metastasis, that is key if you want to make radical surgery and do something good for the patient on survival. You need to remove also the metastasis. And these are basically the scenarios. And then on top of that, it can also be used for watchful waiting. So screening for oral cancer, basically you are looking into, let's say, the tongue. Do you have an optical signal? And then with robotic equipment where with arms you can get into hard to get to locations in the pharynx, in the larynx, so going behind the tongue and down. We can also combine this. It's very difficult to do surgery in these areas, so you need even more the guidance on what to remove and what not to remove. So we can see this going in many ways. The back table imaging of the tumors will basically be pathology while still in the operating theater, while the patient is still on the operating table. And then you get the feedback and say, oh, by the way, it seems like you were not clear of the tumor. You left something behind in this area, and then the surgeon can immediately go back and do the surgery in a better way. This is not possible if this is told to the surgeon 1 week later by the pathologist, because a resurgery is a totally different procedure. So in many cases, it would not be possible and not advisable to go in. So it can really be a game changer for the patient. And in this case, we believe leading to the better prognosis of the patient and improving the surgery and the doing good for these patients. So we are very, very excited and very ambitious about how we can position it in this particular indication. And of course, that's what we are using a lot of time on discussing and talking with key opinion leaders, and of course, in the longer run, also with the authorities, FDA, on how to maximize the benefit for patients through this technology that can be used in so many ways. Also here, there's a lot of immediate potential partnership opportunities. Again, microscopes and open field systems for imaging is used in this area. There are endoscopes, that is when you are going further down, not the tongue, but you're going beyond the tongue. You are typically using endoscopes. And then there's robotic surgery that is coming much into play in this area because it's so difficult to do the surgery in difficult to get to locations. And as you know, this is a relatively competitive area where many equipment manufacturers are now making these surgical robots. And then to see whether you actually removed all of the tumor on the removed specimen, on the removed tumor, there will be back table histology. So you will have basically a scanner sitting in the operating theater, and then the specimens, one removed by the surgeon, it will be taken by the nurse, put into the system, and then there's an immediate readout on you are fine, you removed everything, or no, there's a risk that you left something behind in this area. So a lot of partnering opportunities, well-established companies that are active in this area. So also making it very attractive. Yeah. So the plans in head and neck cancer is to advance FD001 definitely in head and neck cancer, initiate a phase 2 trial, and determine the multiple positioning opportunities in this indication. I went through them. And then the possible partnerships that lie on different levels. You saw all the different types of equipment involved here. And then, of course, the clinical endpoints and the regulatory feedback on that to get an approval, because that's, of course, important that the regulators buy into the endpoint that will be the critical one for getting the label eventually on this product in this particular indication. And we will expand the commercial opportunities and the partnership opportunities, as you could see, quite many in this area. Yeah.
Thank you. Then we are ready to have your questions, Christian, and have the discussion. So thank you for your patience.
Perfect. Thank you so much for that great presentation. Now, we've received a number of investor questions, primarily from Dennis Johansson. And while you've already touched on some, let's start with, obviously, the potentially biggest surprise for many investors was that you choose to combine photothermotherapy with surgical guidance. Now, you already talked about, regulatory feedback, etc. But was potential interest from partners also a factor that made you pursue that route, so to speak?
I can say there's a multiple reason for doing that. I mean, if we take them from an end, we have the regulatory feedback that they would like to see in 12 and 6 months follow-up after fluorescence-guided surgery. And that may actually mean that a program that will run photothermotherapy and fluorescence-guided surgery is not that far from each other. So that's one aspect of it. The other aspect of it is that in the price for photothermotherapy would be in a $5,000 per treatment, per surgery, whereas you have for the fluorescence-guided surgery. And if you take the photothermotherapy price target, then we are up against close to $50,000 per treatment, or even there's been a product in Japan that has been up to $150,000 per treatment. So we are complete different price level. And you can say what was the decision we have to, or the thing we have to decide between were basically, should we add this photothermotherapy that potentially could increase the value with 30 times, and then at worst case risk to delay the fluorescence-guided surgery program half a year. And for us, it was a quite easy decision. And even if I may say that I loved that we get as quickly as we can to the market, we also have to get there with high predictability and with a optimal product. So that was why we did so make this decision. I mean, it's a pretty obvious one if you do the numbers.
Got it. And when it comes to, trial design, you already touched on it, but could you just elaborate a little bit on comparing to a pure surgical guidance trial design, how does including photopharmacotherapy potentially change phase 3 design? And also, is there a possibility to design the trial in a way that you can get an approval for surgical guidance, even if the photopharmacotherapy results would turn out negative?
Yeah, I can start with that. First of all, it will not add many patients to the study, so it will be at a relatively small extra expense and extra effort to include it. The surgeons would like to combine these things because just as we do, they want to do the best for the patients. So combining it would also make the sites that are to participate more excited about it. And then to the last thing, yes, we will make an intelligent design where in theory, if it didn't come out entirely positive on the photothermal part, we would not make the photo-guided surgery at risk. So there would be different endpoints going on the different parts. So you have the full fluorescent-guided surgery study included in the combined study.
Got it. And another question we received is regarding photopharmacotherapy. Can you elaborate a little bit on the steps that are needed to get it phase 3 ready? And are the required costs to get it phase 3 ready covered by the grant that you have received a while back, or will that entail additional costs?
I mean, a lot of the work that needs to be done now is identify a laser system that works with the product, and that we could have offer the entire flexibility, so we can adjust it as to the surgical cavity. So that was one aspect, for instance, that we need to do. We need to have some regulatory work. We need to have some timing work that we need to optimize. But there's a couple of those things that we need to optimize to move to have the best outcome of it. And I think that one way you could look at it is that it's more a question of the optimization than it's a question how well it works, because it will work. We know that from animal, and if you heat up sufficiently, well, you will kill the cells. So the more question, how good could it be actually for the patients? How could we take the cavity? Could we take just larger part of cancer in the cavity? I mean, this kind of question will be. So there, in my view, there's no doubt that we can help patients with this technology. The question is, how well can we help them, and how should we prove it in clinical trials? So in the steps to prepare it, that's what we do this year. We assess how well it works. We will develop the development plan for it, taking it further. And a lot of that work will be partly covered by the grant we have, but also it will be not very expensive compared to well, a lot of other development activities. So we take a lot of risk out of the photothermal therapy program by doing this activity, and that's why we increase the value enormously. So then going into proving clinically, then there was different ways we can go, but we would really like to see this primary work before lock ourselves up on a design. We have multiple designs options we have in mind, but they very highly depend on the outcome with the laser, with the dye, with the dose and the timing. So we would need a little more work before we can disclose an exact design. So we have a couple of opportunities now in our mind.
Understood. And presuming that a phase 3 trial would start in 2025, when can investors approximately expect top-line data from the trial?
It will come to step. The patient will be included relatively quickly. So the patient included will take less than a year, and then there will be some follow-up. So they will come in steps, the result.
Got it. And Now, we've received similar questions for both, the coming phase 3 trial in high-grade glioma, but also the coming phase 2 trial in head and neck cancer. When it comes to potential partnering and then also the associated costs with those trials, I don't know how much you can say, but should investors expect you to partner before these trials and for you to share the costs with potential partners?
I mean, branching into photothermal therapy, one of the questions you had also were if we're due to partnering. I mean, there's in the brain cancer area, there's a few manufacturers, size like and Olympus and a few others, a smaller one, that are present. But there's a very few companies that are present in the microscopes or with microscopes. It's also a market that is very mature. I mean, all those certain hospitals have the microscopes they need, so they're just kind of a placement. If we go into photothermal therapy and we add that to, we can enable new equipment to come into the rooms, and we could provide a lot of value for some of the equipment manufacturers. So suddenly we turn around that market dynamic completely in the brain. And what we see in head and neck, just to give that as an example, I mean, there we can enable new equipment coming in because it's not so much matured or the penetration of the equipment is not so deep yet. So we can enable new types of equipment to have a favorable position and penetrate the market quicker and deeper. So that is the same thing we will be able to see most likely in the brain. Now you ask if we can get the funding of the program, and that's always the question of dilution. We will have dilution by funding, we will have dilution by partnering, and we will take the less dilution that we can get. It's too early yet to say exactly how that will spin out in the brain area for photothermal therapy. For the head and neck, we are pretty certain that partnership is close. But again, I would say is partnership close, not with a lot of money paid in now. I don't think we should expect that. But on the other hand, we don't have any major dilution with them as well. So we get multiple partnership built up, and we think it's more valuable to have a diverse group of partners working on together with us on the indication, rather have one partnership that we completely tied up to. And if you look at on target with the VisuSense, for instance, you look at PhotoCure with the Calstors, you have a lot of examples of slow penetration of presence imaging guided products due to them have been too close to a partner, on the partner do some strategical change that just make it very difficult for them. So we think the diversity of partnership is a very key one. The price we then pay is that the big check is written out later, but we have more security of not being dependent on them. So it's a balance, of course. But we spent a lot of time with partners the last 2 years and talked with many of them, and we think that we get this model working quite well. And also for the partner point of view, of course, it's important that they also see the benefit for it. So we see what we can that's the way we work on partnering now. So multiple partnership, not a big upfront, but not big dilution.
Perfect. And then when it comes to pricing, just so that everybody's on the same page, as you mentioned, implementing photothermal therapy would increase the potential price in high-grade glioma by probably at least an order of magnitude. At the same time, that price may or may not be optimal for example, head and neck cancer. So, having the strategy of different deals and different indications, etc., can you kind of elaborate on how pricing in these different indications will work?
Yes, I mean, probably will work the way that we have the same price for FG001. That could be the $5,000. So everyone can buy it for $5,000. But when you buy a laser and you apply a laser treatment, then you have to buy or purchase that treatment from additional cost up to, let's say, $50,000. So in that way, you can actually both sell the product in presence-guided surgery and photothermal therapy without having problems. And it also will help if you have multiple partnership. Then the price should not, you have difficulty to price differentiate between them. You cannot do that. So it's better to have one universal price for all, and then have the top up of what is applied additionally from the equipment. That could also be the surgical equipment. I would like to say there's a lot of possibility that the surgical equipment can actually enhance the surgical quality by specific features they have. And that's also what we see in interest from some of the partners, that they would like to develop better imaging AI and so on top of on the images that could provide additional benefit and additional value. And the way they can charge for that is that still the price for FG001 is $5,000, for example, and then they actually charge on top of that for the special additional product they provide to the surgical room. So in that way, we actually can have multiple partnerships running at the same time and applications.
Perfect. Then another question that we received about head and neck cancer. There were obviously several possibilities. Can you elaborate a little bit on your decision to run an additional phase 2 trial instead of, for example, combining phase 2b and 3?
Yeah, I mean, this with the phases, I think that is what is important is that we would like to advance it as quickly as possible to approval. I mean, we always said that, and that is our aim, overriding aim. Predictability is also very important, and we could, of course, what you've seen is some company doing phase 3 programs and fail. We don't want to go into that direction. I mean, that is very serious for the company and the investors because there's a lot of money that is missed in that way. So for us, it's very important that we get the endpoint in the trials correct, and that's two aspects of that. The one aspect is, I mean, three aspects. One aspect is toward the regulatory agency. They have to have a buy-in to the way it's the endpoint and the way it's measured. We have the reimbursement, the customers that have to be willing to pay for it. And then we have, and that's the last thing, we have to be very careful that we measure it consistently. And if you look at our product, it's a product that is basically a combination of a surgeon, I mean, a human being, our product that can light up cancer, and equipment that actually can detect it. And the standardization of the way we measure and conduct our trials is extremely important in the sense that if we have to have a robust outcome, this has to be taken care of very, very detailed. So for us, it's better that we are sure that we get the measurement, the right endpoint for the customers, for the regulatory agency, and then it may take a little longer, but that's the de-risking basically. And could say one thing, Christian, if we get it right, I mean, this could be, of course, used for regulatory purpose to support the regulatory, but we do not promise it upfront. That's the way you should see it.
Understood. Another question that we received is regarding large indications, like for example, lung cancer. Could you give any indications for investors when they should expect you to potentially move ahead in a larger indication? And do you think you will require a partner given the probably, for example, larger size of required trials?
I mean, what we think is quite interesting with the head and neck is that in that way we have the diversity of different types of equipment that we can use. And that means it is exposed to a range of different partners that are also relevant for the lung indication. So you can see by doing the optimization toward the equipment in the head and neck and investigating the add-ons you could provide to the equipment, we actually start working on lung cancer as well in the partnering side. But in terms of having a partner in lung, we do not necessarily need to have it for development, but we would like to have it for commercial because the customers are much more spread out. I think another aspect that is important is that OnTarget have launched their product for lung cancer. And what they work on right now in the US is the reimbursement improvement. They also work on getting equipment, let's say, used in the lung field. So, I mean, a lot of this work will take time. And it's not so bad for us to watch and see which segments and how the positioning they do in the lung to use that optimally for how we would advance in lung. Then other large indication, breast, colorectal cancer is also there. But I think it's also important that we take one thing at a time. So we would love to go into all this indication, but we also would like to do it properly. So we don't just have a lot of activity, but do not get anyone further. So the investors should know that we are working hard on lung, preparing that one for partnering later on, but that we will not advance it ourselves at the moment because we prioritize head and neck and brain at the moment.
Perfect. I think those were all the questions that we were supposed to go through. And also, I think this is unfortunately the end of our time. So Morten and Andreas, thank you so much.
Thank you.